Well, good. Thank you, Matthew and the organizers. It's certainly a nice honor to be asked to join for the symposium and to present today. And so I'm talking about genomic selection and precision phenotyping, and kind of think about this in the context of having new tools to do the same trade, the same mechanics of field breeding and improvement. And so we've seen this many times, but just to think about this now in the context of plant breeding and the rate of genetic gain, and with increasing population and production and demand are sort of keeping neck and neck here, and then obviously this is happening on the same acreage. And so the future perspective on this is looking at the current yield trends, and we heard well on this morning, but then how that trend line has to shift upward. And so this is really a combination of plant genetics and agronomy and policy. And so in genetics, we think about this as the rate of genetic gain. So how much better are the populations next year than they are the current year? And so we put this in the context of a breeding cycle. And so this is this iterative process of crossing, making new genetic variation, moving through a long, laborious evaluation process, selecting the best, and then returning those as parents for the next cycle. And so we define the rate of genetic gain as how long it takes to go through this cycle and how much better off you are after going through the cycle. And so when we put that into a nice formula, you look at the components that make up the rate of genetic gain. And so we define this as genetic gain over time. And this is a function of selection intensity, selection accuracy, genetic variance. And so you want to obviously increase the top portion here, and then the amount of time that it takes to go through a cycle of selection. And so this is, you obviously want to decrease that part. And so the components here, the selection intensity is based on how large of populations we can evaluate, and then how many and how few out of that population we select to advance. And so we want to increase this, but it's to a limit because there's inverse correlation with the selection intensity, and that decreases genetic variance if there's too intense selection. Selection accuracy then is obviously how close you are selecting based on phenotype or genomics to the true breeding value. And so we want to increase that. You take out experimental error, you take out measurement error, and you can increase your accuracy. The genetic variance, Ian was talking really well on this, and so if there's no genetic differences in your population, there's not going to be anything to select on. So this you want to increase, but we know this is a mixed bag because I think it's Halauer who's well said that most genetic variance is bad, and so anytime we try to increase genetic variance, especially from wild relatives, most of the variance that comes along is not agronomically favorable. And then years per cycle, this is a big one you want to try and decrease, and this is powerful because these are nonlinear response, so there's a decreasing rate of return as you continually increase selection intensity or increase accuracy, but years per cycle is always a linear return. And so if you have the number of years, you double the rate of gain, always. And so put this then to think about genomic selection and precision phenotyping, and so these are really not new concepts. We just have new tools to go about this business. And so the phenotype is obviously what we eat, but that is results from this complex process involving genetics and the environment. And so these two interact to give us more or less of what we're trying to eat. And so we can work to improve the environment. This is obviously through agronomy, but then there's also stochastic variables in the environment, but we can also work to improve the genetics. And so for this talk, just focusing on the genetics and genetic improvement. And so Borlaug knew this well, and this is what's been done since the advent of plant breeding, is to use field testing and phenotypic observations to observe the underlying genetics. And this was recently brought up by Dr. Baines on my visit to India. This is the original GBS. This is genotyping by seeing. And so obviously if you want a precise phenotype of grain yield, the best thing to do is to plant your varieties and go harvest them. And so this is really the origin of what I would consider precision phenotyping. And so I believe this was obviously also Dr. Borlaug's favorite equation, and he was well familiar with, from what I know, of each of these components here. And so we've heard several times over, just in looking at the scope of the CIMMIT program also and the legacy that continues on, just a selection intensity as evidenced by the massive field testing program and the large population sizes. And so this is to increase the selection intensity. Also the selection accuracy, this is done in the field breeding context, obviously through replication, through multi-location testing, being able not to remove the variance, but to be able to separate genetics from the noise. And so this will then in turn increase your selection accuracy on genetics. The genetic variance, there's many ways to look at this, but I think a simple one in Dr. Borlaug's case was thinking about if you want a short stature wheat and there's no genetic variance in your breeding program, you obviously have to bring material from outside the program and integrate that new variance. And then the big one was the number of years per cycle. And so this is really exemplified by the shuttle breeding program, which effectively cut the breeding cycle time in half. And then if this number is half, you can see that that would double then your rate of genetic gain. And so all of these pieces were working together, and so this is really the context that I think new tools come into. And these components were put into place in the CIMMIT breeding program some 50 years ago, and so it's just not anything quite new. The thing is, we just have new tools to do some of this. And so if you look at field testing programs and now commonly running split plot combines here, and so you can harvest 2 yield plots simultaneously. And so this obviously doubles the number of plots that you can handle in any given breeding program. We also have nice next generation sequencing now that unfortunately wasn't available in the 60s. But this enables us to directly look at the genotypes underlying these candidate wheat varieties. We also have new technologies in remote sensing, and so we can start to use this just as our eye would look at differences between the plots. We can use these new tools, and then also just maybe not quite as obvious, but just tools in data management, barcoding, database, and even computing power to handle these large data sets. And so these are all new tools that really enable the size and the scope of the breeding programs to increase. So in that context, I'm going to talk now about genomic selection and then precision phenotyping. And so really, just on this basic slide to sum everything up, we're trying to predict the total genetic value using these genome-wide markers. And so this would be the same context as phenotyping to look at the breeding value. But we can extract a genotype from a single plant or even a single seed. And so the components of this is develop a training population that's been genotyped and has high quality phenotypes to build a genomic selection prediction model. And then this can be applied on selection candidates having only the genotype and then predicting phenotypes from that. So really the keys and the important components of this is to have inexpensive high density genotypes. And I say inexpensive as a primary driver here because this is in the context of a breeding program, and so the large majority of the material is going to be discarded, and so we don't want to spend money and then throw the lines away. And then another key is obviously the accurate phenotyping, and so really the upper bound of our prediction models is the realized heritability on the trait that you're trying to predict. And so if we have messy phenotypic data going into the model, we can't really expect to have accurate predictions. And so we've been focused on developing genotyping by sequencing primarily as a low per sample cost genotyping tool that can be readily applied in the breeding programs. And so this is really driven by these next-generation sequencing platforms and amazing developments in sequencing output. And so you can see that in this trend line here, starting with the introduction of next-gen sequencing, just this precipitous drop, and so our data acquisition year to year becomes cheaper and cheaper in genomics. We do have to deal with with this a massive amount of data, there's a lot of complex bioinformatics. And so these are reduced representation genomic libraries, and so for a species like wheat with such a large genome, we're still a ways off from routine whole genome sequencing, so we capture only a small portion. And then we sequence many samples together using DNA barcodes, and this makes the per sample cost inexpensive. And so just in that context, I want to briefly mention just some updates on the status of the wheat genome sequencing and how we can then leverage this using GBS markers and genomic selection. And so in collaboration with JGI and using some of these population sequencing approaches, basically have now, and we're working on a whole genome assembly for wheat that's anchored and ordered. And so now we can actually put all of these thousands of GBS markers in a genomic context, and that will give us reference anchoring of GBS markers, which will really help facilitate both the genomic selection and then also association and mapping approaches in the breeding program. And so just the one example I want to give on the genomic selection, and this comes from the CIMMIT program, is prediction of wheat quality. And so these traits like quality, grain yield, are really key targets for genomic selection because these are traits that are expensive to evaluate, and they're typically evaluated many years into the breeding program. And so this gives us opportunity both to save cost and to accelerate the time to making selections. And so this was done on the International Bred Wheat Screening Nurseries, and there's quality data for milling and baking, and use genotyping-by-sequencing markers on these. And so this is sort of a cross-validation just to get an estimate of the prediction accuracy in these studies. And so what you see from this study is actually across these range of traits, there's fairly high accuracy, 0.5 to 0.7. And so this gives us good indication that yes, we can use these whole genome markers and actually get a decent prediction for these complex traits, even something as time-consuming as assessing a low volume. And so with this, then, we're integrating in the entire scope of the CIMMIT Global Wheat Program to really implement and evaluate the utility of genomic selection in a big way across the program, and then also extending this in the target regions of South Asia. And so this is a project we have now supported through USAID and in collaboration with Kansas State, Cornell University, and CIMMIT and the BISA, and then the national programs in Pakistan. And so with extended yield testing and evaluation across these 4 sites in South Asia, and then integrating genotyping basically on the entirety of new material coming through Ravi's program, we'll have over the next several years really large-scale testing and evaluation of implementing this approach in the CIMMIT bred wheat program. And so sort of the conclusions on genomic selection and thinking about how we can address each of these components and accelerate the breeding cycle. This is really a tool to enable selection on single plants or single seeds. And so to think about doing a bake test and having to have a certain amount of seed, we can actually make this prediction from a single plant early on in the breeding process. This also is really enabling selection in unobserved environments. And so we can make prediction models across different environments, different target locations, even though earlier on in the breeding process, prior to international nursery testing, you can actually get a prediction before sending lines. This also didn't speak about, but gives opportunities for maintaining genetic diversity. And so we can actually maintain this by using the markers to favorably combine and favorably select lines that maintain rare and low frequency alleles. And then also this enables evaluation of larger populations. And so a good example here again, going back to the bake testing, is that the baking labs are really limited by the number of samples that can go through. And so we can easily genotype tens of thousands of samples for a relatively low cost. And this can greatly then expand the number of lines, basically, that we're able to do selection, say, for bake quality. So now I'm going to shift and talk in the context of high-throughput phenotyping, and really to define this as automated or semi-automated platforms for rapid precision assessment of the traits that we're going after in the field. And also just to note that this really needs to be high-throughput analysis. And so in the context of these large populations, if we spend more time analyzing the data than collecting it, it doesn't really become a high-throughput approach. And we really want, in the context of the breeding program, again, to decrease the phenotyping burden and increase the efficiency of the breeding program. And so if this phenotyping approach, is high-throughput phenotyping, doesn't actually increase the efficiency, then here again, there's not much point. And so I really view this as a multidisciplinary approach, really at the intersection of physiology to know what we're looking for, what we want to look at, plant breeding genetics to understand the populations and to really apply this, and improvement, engineering and bioinformatics to make these complex systems work and to utilize the data coming out of them. And so just to give you some examples of some ways we're going about this in our group is we're using what we call Geo-referenced proximal sensing. And so this is the integration of precision GPS. These are RTKs, so they have a real-time correction. And so these are centimeter level accuracy, easily placing measurements within a plot in the field. And then different arrays of sensors that can then be geo-referenced, each data point back to a field plot. And so here's just an idea of the platform configuration. Here's an example of a phenotyper we have with 2 GPS units. And so from this, we can know the angle and the orientation of this toolbar at all times, and then different sets of sensors that will move over the crop canopy, taking measurements on plant height, NDVI, canopy temperature, et cetera. And these are sampling about 10 times per second. And then each one of those data points will be referenced with a GPS coordinate. And so here's just a cross-section example of that phenotyper kind of output on the latitude and longitude of some data points, and each one of these individual points would be a single measurement. You can see here turning around at the end of the field and then moving into the field plots here with the left and right GPS, and then the extrapolated position of each one of those sensor measurements as you move through the field. And so what this looks like then on a field level and some of the raw data, this would be a field of about 50 by 20 ranges, so around 1,000 plots. And you can see the ranges going this way with these low values being the bare soil between ranges. And so you can see field variation, kind of gradients across the field. Here would be bare soil at the end of the ranges. And then, but you can also see differences between the plots in the field, this being due to genetics. And so to processing this type of data is going from the raw data, defining the plot boundary coordinates, and then from there trimming the data, and then based on the row column design of the experiment, being able to assign all of these data points to a genetic entry in that experiment. And so just an example, this would be an example of using sonar to measure plant height. And so this would be just a cross-section moving down one column in the field, but you can see measuring basically the distance from the boom to whatever the sonar touches first. And so this would be measuring one canopy here, touching the bare soil between the plots, and then measuring the next genotype, the next canopy here, and so forth. And so the thing just to point out is that if you just take a simple average of these different measurements, we have basically significant differences between each one of these, being able to measure centimeter level differences in plant height. And so what this looks like, we did some comparisons here of basically going out and measuring these with a measuring stick and comparing that to the phenotyper. And so these are each one of these dots representing a single field plot, and then looking at assessments of the phenotyper on 2 different days. And so you can see here that across 2 different days, this would be on the 31st and then the June 6th, and you can see that there's a pretty high correlation between 2 dates. And so any variance here, obviously because these are the same plots being measured 2 times, would be sort of experimental error. And this actually turns out it's considerably more accurate than any 2 people. So here's, we went out and measured plant height just with a measuring stick, and so you can see the correlation between the phenotyper on 2 different dates is actually much higher than 2 different people measuring the same plots on the same day. And so really the conclusion on this was that we are getting much more precise measurements, and we're also being able to measure much faster. This phenotyper can measure, we're measuring 3 plots at once, but on average you can measure a plot every 1 to 2 seconds. And so the other conclusion on this was I looked at this and said, hey man, I'm more accurate than 2 of my students here. But they looked at it and they concluded that I should go do all the plant height measurements because I was more accurate. And so what this also enables is because of this rapid assessment, we can start to look at multi-temporal time series that have previously often been intractable just due to the sheer burden of assessing the field over and over again. And so here would be that same field measured at 3 different points from heading through grain fill. And so you can see sort of field level maturity here. You can also see the field variation, but then you can also see differences between plots and genotypes through that field. And so this enables those repeated measurements over the time. And what we can then start to do is identify dynamic differences among genotypes. And so this is just a subset of entries from that trial. And you can see that at heading, this is NDVI, so a normalized difference vegetation index. And you can see that early on in the season, they basically start close to the same point. And then at full maturity, they basically end up completely dried down. But in between there's really dynamic differences in how they get from point A to point B. And so we can start to look at this and actually start to think about breeding and making selections for something like grain fill duration. And so we're also looking at basically the same approach but different platforms. And so this is Jared, one of the students in my group, basically with a handheld version of this phenotyping platform, integrating the GPS, infrared thermometer, green seeker. And this is being used a lot now with Matthew's group for trials here. We can also take this approach to some of these unmanned aerial vehicles and be able, more on a lower resolution, be able to start look at the field as a whole. And so to sum up on the high-throughput phenotyping, this really enables more precise assessment, evaluation of larger populations, and so really increasing this selection intensity and selection accuracy. What do I have, 2 more minutes? Oh, okay. I'll hurry up here. And so I just want to conclude that precision also comes from reducing errors. And so just thinking about this in the context of these many steps that we go through in this evaluation process. And so even if there's a 1% error or any type of errors entering in the process here, at the end of this process, you can have quite a large amount of basically random data. And so that's obviously going to decrease your selection accuracy. And so with this, just to think about really the need for implementing apps for electronic data management to reduce errors, increase speed, and really make the program as a whole more efficient and more accurate. And so this comes into the concept of what we like to think about as one handheld per breeder, enabling precision phenotyping in the sense of accurate data management and recording. And so I want to conclude on this and just think about this in the context of turbocharging the breeding program. And so to implement genomic selection, high-throughput genotyping, you can think about this nice engine right here, and these are turbochargers on here basically to make the whole system run more efficiently and faster. But if this engine isn't firing on all 8 cylinders, then there's really not a lot of use on adding a turbocharger to an engine that looks like this. And so the mechanics of the program must be functioning well, and data management is really key. And so that has broad implications, I think, for depending on if you're in the business of funding research or you're a student, or if you're in a breeding program, how to do this. And so acknowledgement, just to thank the students in my group. And Sarah Battenfield is working on the quality predictions with Ravi Singh, and Jared Crane is here working on the high-throughput phenotyping. Acknowledge that this work is supported by USAID and the Bill and Melinda Gates Foundation, the McKnight Foundation, and Kansas Wheat. So thank you.